All right, thank you for this nice introduction and for the opportunity to present our work. I'm going to present an overview of the work we've done in our lab for a few years now about the advanced treatment of micropollutants in wastewater treatment. And we studied a lot of different processes, from adsorption processes and more recently oxidation processes, and in relation with disinfection. So I'm going to show you everything and the history of what we've done since a few years. So as you know, organic micropollutants are present in wastewater due to various activities. And in the end, they go to the wastewater and to the wastewater treatment plants, in mostly where we can analyze these micropollutants in France. Some of them are removed by the treatment in the wastewater. For example, mostly hydrophobic volatile compounds or biodegradable compounds are removed by the adsorption in the sludge, for example, or by biodegradation with the process. It's, for example, PAH are well removed, but many micropollutants are not removed because they are more hydrophilic, such as pesticides or pharmaceuticals, and they can be released and discharged in the rivers, and they pose some potential threats on the ecosystems. So in France, in Europe in general, we have several regulations that um, can limit the production and the discharge of micropollutants in the rivers. So there's the REACH regulation to limit the uses of micropollutants. In France, we have such plans also to reduce the emissions of micropollutants to the water bodies. And it's not exactly in Europe, but we have the example of the Swiss regulation that for a few years now they did this regulation to remove 80% of several pharmaceuticals, mostly molecules, in wastewater to avoid their impacts on the ecosystems. So there's a list of 12 micropollutants, and the regulation says that you have to remove 80% of 6 molecules among those molecules. And so based on this regulation, there is an ongoing discussion, ongoing regulation in Europe that will be apparently very close to what the Swiss have done. And the goal will be to the same, and to limit the discharge of micropollutants, and it will necessitate to add additional treatment in wastewater treatment plants in a few years. So that's the context. We will probably get there soon in Europe. And so we have a few technologies available to treat micropollutants in wastewater. And these technologies have been used in drinking water for a long time now, such as oxidation processes with ozone, or more advanced oxidation with radicals produced with UV and peroxide, for example. There is also the adsorption processes with activated carbon, or even with zeolites. So these are mostly for adsorption. And in the end, membrane processes, either with reverse osmosis or nanofiltration. So back in 2012, we started at LESU to work in a project with the French operator of wastewater treatment plant. We are very close with them to work for a few years, and we've done several PhD projects with them, and they started to ask us if we could work with them on the adsorption on activated carbon. And they installed a big pilot, which is a Carbo Plus filter like this, with fluidized activated carbon inside. and it's built by a French company called SOR. There was a first PhD thesis, Romain Maillet, who studied the adsorption of several micropollutants, mostly pharmaceuticals, by this big pilot. And they found that this pilot was able to remove with good removals a lot of compounds, mostly alkyl phenols, bisphenol A, parabens, and pesticides. But for some compounds, there were quite intermediate removals, and some molecules were also recalcitrant to the adsorption. So, yeah, overall, this process has satisfactory removals, but some limited performances for specific micropollutants. So this is where this pilot was located. It's on the Seine-Centre wastewater treatment plant. It's one of the most important wastewater plants in France. We're treating wastewater for around 1 million inhabitants. And it's so here you see the scheme of the plant with the primary treatment and biofilters for biological treatment, and finally denitrification and discharge in the river. And this pilot was located just before the discharge. And it was not treating the whole flow of the plant, because it's huge. It's 2,400 cubic meter per day. And the Carbo Plus pilot was only treating 860 cubic meter per day. In this study, we've done a lot of analytical campaigns during a few years with almost 50 organic micropollutants targeted. And we sampled also some water inside the plant at the different treatment steps, and of course in the pilot and at the effluent of the pilot. So we've done a second PhD thesis on this topic to continue working on this treatment. And in the end, we detected only 28 micropollutants among the 50 that were targeted first. Mostly pharmaceuticals, but also pesticides and some hormones and PFOS. A few results of these 28 micropollutants. So as it was shown in the first PhD thesis, there was a good removal for a few compounds at the carbon dose that was used in the pilot, but a few molecules were also recalcitrant. So it's always the same. There was sulfamethoxazole, PFOS also, and clarithromycin were always with low removals, and some other molecules were always well removed, such as the pesticides diuron or benzotriazole. What we've done in this PhD to go a bit further was to compare the proportion of molecules that were removed in the conventional treatment, so in the wastewater treatment plant, by the biological treatment mostly, and the removal by adsorption on the activated carbon. Because we had these removals on the pilot, on the carbon, but we didn't know if it removed most of the molecules, or if the molecule was maybe removed mostly on the biological treatment. And so this graph, you can see that there is a lot of differences between the molecules. For example, some of them, like acetaminophen, are almost entirely removed by the biological treatment. It's a removal of, in yellow here, around 98-99% in the wastewater treatment plant. And the adsorption of activated carbon is just a bit more. For some other molecules, like here tetracycline, it's only blue. It means it's not removed at all in the biological treatment in the wastewater plant, and it's only removed by adsorption on the pilot. So here, whoops, sorry. So here the pilot has a big interest for this type of molecules. Same for diuron here. And for many molecules, there is part removal in the wastewater plant, and the pilot is added another removal on top of this, and we can reach satisfactory removals thanks to this complementary between the conventional treatment and the adsorption on activated carbon. So you see here that when you sum the removals from the plant and the pilot, that we can reach for many molecules the 80% that are targeted by the Swiss regulation. In this PhD thesis, we've done a lot of lab work also to try to understand the complex interactions that take place in the adsorption process. And so we've done a lot of different studies with dissolved organic matter to try to understand the interactions between micropollutants and their properties and their chemical properties and the adsorption on activated carbon, the properties of the activated carbon, all these that are written here, and the properties of dissolved organic matter. And we found quite interesting features that molecules can form complexes with organic matter, and this can help to adsorb the molecules in some cases, or in some other cases it can on the contrary be more difficult to adsorb. So we had a bit more of an understanding of how this adsorption in the process works. So after this adsorption process, we started working on oxidation process in the same PhD thesis with an ozonation pilot that was installed also in the same treatment plant. So here you see the same process of the plant. And in this wastewater treatment plant, we have the opportunity to access pilot plant, pilot site where we can install a lot of different pilots. And we installed this ozone generator here to treat some of the effluent of the wastewater plant. So we started working on the same molecules and tried to check the different specific doses of ozone that were that should be used to remove the molecules. So we found out that there was a good accordance of our results with the constant, the reactivity constants that were available in the literature. So here, all these molecules were classified depending on their known reactivity, but, except paracetamol, I think. Yeah, so these were the most reactive, and some other were less reactive. And the only one that was surprising was acetaminophen here at the far right. We don't know why it was less reactive than expected. And we showed that the concentrations, the specific doses needed to remove 80% of the molecules was at least 0.33 gram of ozone per gram of organic carbon. After this, we grouped, first we grouped the molecules in different groups of reactivities. So here in the group A, you have the most reactive molecules. Group B is intermediate reactivity, and group C are the less reactive molecules that need to we need to apply a more higher specific ozone doses to remove them at satisfactory levels. And if we look here at the bottom, if we look at the average removal at each different doses that we tested, we found out that to reach again this 80% removal, we need to apply supply at least 0.4, 0.5 gram of ozone per gram of organic carbon. And what we've done next was to couple ozonation to activated carbon to try to remove molecules that were either not well removed by ozone or not well removed by activated carbon adsorption. And so we tried to combine the two processes to see if we could, in the end, get satisfactory removals for these molecules. And we found out several interesting results. So these graphs are not really easy to understand, but they show that where there's an arrow here, they show an additional removal thanks to one of the two processes. So on the top graph, you can see that there is an additional removal when you add activated carbon on top of ozone. So for these molecules like atenolol, PFOS, ketoprofen, there's an interest to use adsorption on activated carbon after ozonation to remove more of these molecules. And on the bottom graph, it's the contrary. It's molecules that are poorly adsorbed on activated carbon can be more eliminated thanks to an additional ozonation. So for example, diclofenac here is poorly removed by adsorption, but if you add ozone, it will be well removed and you can reach 90% removal after all these the two processes. So there's a good complementarity between the two processes, and the ozone and adsorption on activated carbon compensate their removals. After this PhD thesis on ozone and adsorption on activated carbon, we changed the topic with a new project, again in collaboration with the SIAP operator of water treatment plants, because there was a new topic for them. It's to make the Seine River swimmable for the Olympic Games in Paris. So they installed you see here the installation, the process with performic acid. It's the molecule you can see here. And they installed this upstream of Paris in a big wastewater treatment plant we have near our lab, the Seine-Amont wastewater plant. And they also worked on bench scale trials in another wastewater plant. And so we started working on this performic acid. It's a new emerging oxidant, and it's mostly used for disinfection because it's efficient for disinfection, and it will be used to reach a good quality of the Seine River for the Olympic Games. And you can form it from formic acid and hydrogen peroxide in acidic conditions, and you get this molecule. It's been patented by a Norwegian company. The thing is that this performic acid molecule is very unstable. So if we performed a kinetic decomposition, and we found out that it's almost totally removed in less than 1 hour. The good thing is that since it's unstable, it's not staying in the river, and it's decomposed mostly to carbon dioxide and oxygen. So in the river, it's good to protect the ecosystem in the end, but for the disinfection and the reactivity, it's important to know this decomposition. So since it was interesting for disinfection, we started asking if it was also interesting to remove or at least react with some organic compounds in the wastewater. And we started working on oxidation kinetics of pharmaceuticals to see if this oxidant could remove some of them. And we found out that it was very reactive with some molecules, such as ranitidine here on the left. There was a total degradation in a few seconds. And some other molecules like lidocaine here is also well reactive with performic acid. Some of them are less reactive, but still furosemide can be a bit eliminated. But other molecules, you see the list on the right here, carbamazepine, diclofenac, these molecules are not removed at all by performic acid. So we've done a lot more studies on this reactivity of molecules, and we found out that these specific sites, specific groups of molecules were well removed with the thioether, the sulfur atom here is attacked by performic acid, and tertiary amines in yellow here, they are also attacked by performic acid. And to some extent also the furan group. So there are specific groups, molecular groups that can be attacked by performic acid, and that explains the reactivity of these molecules. But in other molecules, there's no group attacked, and so there's no reactivity. So it's very selective, very with specific groups. So we've performed next similar reactivity studies, but directly in the wastewater by spiking wastewater effluents with the organic molecules. And we found out that it was in accordance with the reactivities that we found in pure water in the lab. But the degradation of some of them, for example, lidocaine here, was quite low because there was also competition with the water matrix. And also in the wastewater, there's a the decomposition of performic acid is much faster, so it's not available to react with the molecules anymore. But some of them, like ranitidine, you see, you can reach almost 100% removal. To understand more the mechanisms of decomposition in the wastewater, we performed high-resolution mass spectrometry to try to detect degradation products. So here you can see a graph showing all the molecules detected before and after disinfection. So there was a lot of molecules removed, but also some of them were new, and we tried to identify some of them. And what we found out in the wastewater is the formation of class of molecules called N-oxides. And for lidocaine, I showed you just before that it was very reactive. We found that it was decomposed into lidocaine N-oxide, and we confirmed this with analytical standards and specific analysis of these molecules. So when we discovered that, we tried to understand more how it was formed. So we've performed different experiments of degradation of lidocaine by performic acid, and we showed that it's almost entirely forming lidocaine N-oxide. You see, you keep the same concentration, but it's transformed into the N-oxide. The thing with these N-oxides is they are not biodegradable, and they are very stable, so they stay in the system and they can stay in the river. It's not really clear if they are toxic or not, so there's more work needed to understand more these byproducts. So yeah, we published these mechanisms of transformation. After we found out that PFA, performic acid, was not really reactive, we asked ourselves if we could make it more reactive, for example, by coupling it with photolysis with UV to form radicals. And we performed several experiments, again, kinetic experiments with molecules. And this coupling here in green, you see the reactivity when you couple UV photolysis with performic acid, and it's much more reactive than you cannot even see them in blue. It's performic acid only, and in orange it's with UV H2O2. with UV peroxide. And so UV PFA is even more reactive than UV peroxide or UV alone or performic acid. Yeah, that's it. And again, same as PFA, it's very selective because, for example, carbamazepine is not removed at all, and acetaminophen is very well removed by this advanced process. So to understand how it worked, this process, we tried to analyze the radical species by electron paramagnetic resonance. So I will not go into details of how it's formed or how it's analyzed, but you have to use a trapping molecule. And you can, with this graph, you can check the different types of organic radicals that can be formed, or radicals in general. So in yellow here, there's the typical signal of hydroxyl radicals that are formed, for example, during a UV peroxide process, but also here you have also the other signal linked to organic radicals formed specifically from performic acid. So there was a complementarity of these two species of radicals that allow this process to get more higher removals. All right, so that's all we've done in this last PhD thesis, and we found that, as I said, that there was a overall low reactivity of performic acid, but it's really very selective with specific molecular groups. There's overall a very low formation of degradation products. I showed you these N-oxide products, but in the end, it's not very important in terms of number of molecules. And we found out that the advanced oxidation with UV is much more reactive. An interesting thing that we found is when we grouped our molecules in groups of polarity, of hydrophilicity, here you see this graph, the molecules that are the most removed by performic acid are these groups in green here that have the lowest hydrophobicity or the highest polarity. So it's interesting because, as I said in the beginning, molecules that are removed in the wastewater plants are usually the most hydrophobic because they are sorbed on the sludges. And on the contrary, performic acid is removes more of the hydrophilic compounds. So it's interesting as a tertiary or advanced treatment of micropollutants because it's complementary as compared to the processes in the wastewater plants. So what we are still ongoing, what we are still doing is to try to determine the different degradation products that are formed during degradation by PFA or by the advanced oxidation with UV. Since UV performic is much more reactive, it forms also more byproducts, and so we will try to identify these byproducts. All right, so that's all for my presentation. I'm gonna thank you for your attention. Thanks to all the staff and teams in different labs where we had the opportunity to work with, and the three people who did most of the work, the two PhD, Ronan and Christelle, and the postdoc also who has done a lot on performic acid, Maolida. And thank you for your attention.